that we organized in a topic related to biology, and we are very excited about it. So today, virtually with me, I have Dr. Carme Cortina. Hello, Carme.
Hello, good afternoon.
Hello. Carme has a PhD in biomedicine by the UPF. She is a research associate at the group of colorectal cancer at the IRB in Barcelona. She has experience in the use of Crispr Cas9 system that we are going to talk about it, and is specific in the study of tumors. And in fact, she's one of the persons involved in the subject of molecular biology at the degree of bioinformatics, and she is responsible she was responsible of the part of CRISPR this year. With us today, we also have Janic. Hello, Anna.
Hello.
Anna did a PhD also in biomedicine at the IRB, also in Barcelona. She's a tenure-track professor at the UPF in the Department of Medicine and Life Sciences, and she is leading a research team that also works in cancer, more specifically in tumor suppressors, and in particular with the P53 gene. And she is the responsible of the molecular biology subject at the bachelor degree of bioinformatics. And finally, we have also Dr. Alberto Meseguer. Hello, Alberto.
Hello, everybody.
Alberto is a doctor in bioinformatics by the UPF, and as myself, he is academic coordinator of the bachelor degree in bioinformatics, where he teaches various subjects, including biochemistry and structural bioinformatics. Okay, so let's start. Let's start with CRISPR. So this is a technology. I think we have all heard about it, which was discovered about 10 years ago. And the discovery is credited mainly to women, Emmanuelle Charpentier and Jennifer Doudna, who won the Nobel Prize in Chemistry for it in 2020 for the groundbreaking work on CRISPR technology. So let's start from the beginning. So Carme, what is CRISPR, and what does it mean actually, CRISPR?
Yeah, so CRISPR basically is a genome editing system. So what this means is that this is a tool, a genetic tool that allows to introduce, to change, to modify the genome of any given cell, even a human cell, adult, embryonic, of other organisms, and so. So the revolution of this genome editing system is that it's much easier and cheaper and easier to use than previous editing systems that were existing before the CRISPR was discovered. So CRISPR is not a unique and only way to modify genomes. It's just that it's so elegant to design, so easy to use, so cheap, that it has made the technology of genome editing available for almost every lab, which a basic knowledge on molecular biology, let's say. So this has revolutionized the field because many labs now can do genome editing, and now experiments that were not possible before, they are possible. So that's the main revolution.
Great. Also, well, there are many places to start a story, but one possible way could be the University of Alacant, right?
Of Alacant, yeah. So in Alacant, so everything, CRISPR, the acronym is an acronym. That means clustered, regularly interspersed, short palindromic repeats. Sorry. And this name was put by a Spanish scientist called Francisco Mojica, that he was in the University of Alacant, but like back in the 90s, studying biology and genetics of bacteria. And he realized that many bacteria, like 50% of bacteria, contain very mysterious, short, repetitive sequences in a precise location of the bacteria genome that he called CRISPR, because they were clustered in regularly separation or interspaces, And these repetitions were breaked by variable sequences. And then he hypothesized that this could be an immune system that the bacteria had against invasion or infections of virus that infect bacteria. And so bacteria, this cluster of this genomic cluster, hypothesized that this could be this immunity of the bacteria. The thing is that he didn't receive much credit because he didn't figure out how exactly this system worked, how this was, if this was an immune system indeed. Indeed, he was right. So his hypotheses were right, but it's just that he didn't proceed to develop this as a genome editing tool that can be used. So what Doudna and Charpentier did was to dissect which were the mechanisms that the bacteria had and purified the minimum components that you can have in a tube so you can edit a genome in a tube. So that's what it did. So I don't think I explained yet what CRISPR does as a genome editing thing or how the bacteria use that as an immune system. So what it turned out is that the CRISPR is a complex of a nuclease or a protease called Cas9. So it's a protein that can cut the DNA in a precise location. And this location is guided by another part of the complex, which is an RNA called guide RNA. So this RNA can recognize specific sequences that you can design at will. So if you say, I want to edit this gene because it's a gene which is very important to knock out, I want to study this gene. So you can design a guide RNA and then you complex with Cas9, and then this is the minimal components that you need to deliver. So this CRISPR-Cas9 complex can cut the DNA in that precise location. And then what happens is that the cell repairs this damage. The cell itself repairs this damage by different means. One mean is that it chunks and adds some molecules, some base pairs, like randomly, and this creates some variation and frame shifts, so the reading frame of the gene is disrupted. So this, in the end, turns out in a premature stop codon, and the gene is lost. So bacteria does that in the virus. So bacteria in these interspaces that Mojica found, these were guide RNAs that the bacteria inserted in their genome, and these guide RNAs were sequences from virus. So virus that had infected the bacteria before, the gene kind of copied a bit of that sequence, and it had it already knowledge. So if the bacteria faces a second infection by the same virus, it can now form this CRISPR-Cas9 complex and cut that DNA and destroy that virus. So it's protected against more infections of this virus.
Carme, could you explain us a bit more about these sequences from the virus? I mean, how long are they? And is there a reason why the bacteria decides to store a particular sequence or another?
So yeah, they are very short. In fact, there are 20 base pairs. So it's long enough to be very specific, so that not being repeated randomly in other sequences which are not the precise sequence, but short enough to be able to insert it, to produce a complex which is not very big, and also it's very amenable to translate into a tube genome editing tool. So the repeats here in this graph, which are in the locus, CRISPR locus, are depicted in orange, I see. So then this is translating to the tracer RNA, which is like the fixed part, like a scaffold for the CRISPR RNA. So this, in the end, it's like 100-something base pairs, but the variable part is just these 20 base pairs. Which sequences the bacteria decide to integrate specifically from the virus, I think this is not very well studied and not very well known and might be random. But there's just one, the only limitation is that the sequence has to be followed by a limitation in the sequence constraint, which is called PAM sequence. PAM sequence is proto-adjacent motif, and normally it's three nucleotides only. Normally it's a nucleotide and then guanosine, GG. So every sequence that is preceding two Gs or if it's in the reverse format, it would be C to Cs. There's a guide there that you can use, the bacteria can use, or scientists.
Okay, thank you very much. I think this was a very good introduction. So, but this year you started using CRISPR in the subject of molecular biology. So you teach the students about the technique and also how to do it, which is quite impressive because this is a first-year degree, a first-year subject.
Yes.
Sorry. So the students didn't have a lot of previous experience in the lab. They don't know much biology by that moment. So could you tell us about how was the experience? I mean, what did they do? How was it?
Yeah, so the fact that the first year students can use CRISPR or can generate these tools, students, as you say, that maybe they were like first time touching pipettes, and so it demonstrates that the technique is like very easy to use. So these students, what they did is like with 1 week time, they generated a specific CRISPR tool. So they inserted in a plasmid a single guide RNA towards a specific gene of interest. In their particular example, it was P53, which is a well-known tumor suppressor. So the aim of the practicum was to generate a CRISPR-Cas tool that could knock out P53 gene. So what they did, in fact, was to create the tool of the CRISPR-Cas9. So there's several ways to deliver the tools into the cells. The most probably used is to use a DNA plasmid, like a circular small plasmid, in which there's already encoded the sequence for the Cas9, which is a fixed part of the complex. And then you have to clone your specific guide RNA towards your gene of interest. So this is what they did. So the second part of the CRISPR experiments, which can take longer, is that then you have to deliver these tools into the cells, and you have to validate that the cells have the correct edits, and then perform whatever experiment you want to do, like when you knock out the gene versus the control cells, which is different, to figure out the function of the gene, for instance. So this was maybe out of the scope for 1 week starting students to do a practicum, but we covered all the steps for a CRISPR experiment in depth in the theoretical lessons. So I must say that I was impressed. I have a very positive experience, and I was impressed because they were like very excited for the potential of CRISPRs. They were very curious, putting questions and asking themselves lots of things related to the technique. And well, I think I hope I could transmit my enthusiasm toward this, because I think it has a lot of potential also for them, because even if they are bioinformaticians, and in the beginning I was thinking maybe they are not interested in all these handling, and they did, in fact. And it's true that there's a lot of field for bioinformaticians in the CRISPR field. There's a lot of needs, because there's then websites for design that can be like further developed or improved for analysis of the edits. There are CRISPR applications, which are like CRISPR screens, in which then you require complex analysis of the genes affected when you knock out the full genome in complex experiments. So I think that I was trying to encourage them that you must learn this, and because this is a must for you.
Yeah, I can tell that the students definitely enjoyed and felt your enthusiasm.
I hope.
And they had heard about this technique, so they were very, very happy to be able to. Even if it's not a full experiment, it's just the beginning, right? But it was very fascinating for.
It's very fashionable now. It's true.
Yeah. So I would like to ask to Anna, because I think I remember that you told us the next year I would like to introduce CRISPR to the subject, and I have a person that will prepare it. So Anna, why did you decide to add CRISPR to the subject, and I mean, is it something that appears in many programs, or?
Well, actually, I'm very happy that you asked me this question. To introduce CRISPR, I was thinking not only that it's fashionable and something that is completely new, it's only that it has a real biomedical and bioengineering applications. And I really think that nowadays the medicine will go forward with CRISPR. It's something that is really revolutionary. So before answering why it's so revolutionary, what I can tell you to the answer to your second question, which is like, I don't think anyone in the UPF does CRISPR. So we are actually kind of pioneering this together with Carme and with other students in the lab. We are pioneering this, introducing to the undergrads actually the CRISPR and how on-hands CRISPR. Although what they do is the theoretical, okay? In many subjects, this is theoretically, they know, they watch Netflix series as well about and so they know how it works. But on-hands experience, I think, is the first one that we have implemented successfully, I would guess.
Good.
So, and then the other thing is, about the application to basically to biomedical engineering. I just want to say why it's so revolutionary. So it's because before, for example, it was known that, for example, to make a knockout of any cell line was possible before, but it was not easy as it today. But this was, for example, the in vitro technique. But just to recognize that now for studying the gene function in vivo, the CRISPR make it so different, right? Because to study the gene function in vivo, you basically need to make the knockout animals, which can be zebrafish, Drosophila, mouse. In case of mouse, we needed to wait like a year maybe to get the knockout mice, because you need all these complicated vectors, the cells, which are the embryonic stem cells and everything. But nowadays, with CRISPR, it has been so revolutionary that actually the companies are now selling the knockout mice, which you can get, maybe you just order your favorite gene, your favorite mutation, even in very like single mutation, you can make it and you can have your mouse in 2 months, which is really revolutionary. And this kind of speed up a lot our researchers, but not only that it speeds up, but also it makes it more cost effective. Right? Because before we need to breed the mice for a year and all of this, but now it makes it more rapidly and more efficient. And then the other thing that we are using is the 3D structure, but this is Carme. We can brought this for the round table, which she was also pioneering how you can make the CRISPR inside of the organoids, right? And how you can make your knockouts of the favorite genes that are so important in some diseases like cancer or some neurodegenerative diseases. And actually you can use this, what we call the 3D structures of the cells, and we can use them and put the therapies, which nowadays they can be directly related to the patients that are in the hospital, and this can be also be treated. And then something what Carme mentioned that was revolutionary in the field of bioengineering is these CRISPR screens, right? So this is like if you want to study your favorite process, that is like has so many, like cancer, for example, that has so many pathways, different pathways. You can knock out your favorite genes or overexpress them in just of one cloning. Like cloning, what we call like one experiment, right? In one vector, we can have all these guide RNAs, they can be thousands. And we have 2 different types of the screens, right? So we can have these positive screens, which we can pull all the guide RNAs all together, infect them not only in vitro cell culture, but we can do them in these 3D structures that we call the organoids. And also we can do them in vivo, right? So we can do it in the whole animals. That can be zebrafish, mouse, of everything. And we knock out like a panel of all the genes of interest, and we follow how the cells react to each of guide RNAs. And Carme already mentioned that bioinformaticians are really needed for this, because to identify which of the guide RNAs gets like in positive screens, mostly like upregulated, we really need them to identify.
Definitely.
And then we have other types of the screen, which is kind of opposite what we do as well in the lab, is like the negative screen. So we can put all these guide RNAs all together in one vector, right? It's so simple, as Carme said, simple cloning, everything put inside of the cells. And we can see which cells up on those guide RNAs, so those genes that are down, we kill those cells. For example, in cancer, this is so important in the terms of therapies because you actually can, in a couple of experiments, you can identify how those cells are vulnerable to those genes. And this really made our life as a scientist in biomedical engineering so much easier, to make us to explore many different areas, how we can do it. So, yeah.
So do you see that this technique is now incorporated as a routinary tool in all molecular biology labs?
Yes, absolutely. Absolutely. Yes, absolutely. Especially because nowadays it's so spread around and it's so easy and so cheap that you can really get this construct, and cloning is what Carme said we can do with undergrads in a week. We can clone it. It's true that after that you put in the cell, it takes some time. But yes, absolutely has revolutionized our research. Yes.
Okay. So yeah, for sure. I can appreciate the value in getting new knowledge, right? In designing new experiments and all this. And then I wanted to ask you, in the medical in the medical practice today, how is CRISPR used? How far have we gone?
Well, actually, there is, I can tell you, but I've been done some research that apparently nowadays there is around 20 clinical trials that are basically using as a CRISPR tool that we can use for the clinic, for the therapies, in the clinic. And one of the first one that has been FDA approved was the phase 1 clinical trial in 2018. And it was of course, in the US was approved. And it was basically is how we can use the CRISPR to edit the T cells, the T lymphocytes. And we can make them more more prone. We can express on them specific what we call antigens, okay, that are more efficient for the cancer immunotherapy. Cancer immunotherapy nowadays is working extremely well in melanoma, in many types, like lung cancer, in many types of the solid cancer. So this was really important. And basically what they do in the clinics and in this clinical trial that was already approved is that they take the lymphocytes basically from the patients and they can modify them ex vivo, and they can put them back inside of the patients. And what this helps to those patients to be more reactive to the immunotherapy, which is basically now in the PD-L1 mostly used, the immunotherapy that has been already the PD-L1, anti-PD-L1 has been approved as well as a drug. So this was like one of the therapies that has opened the door. And then after that appear a few other ones. So there is this one, which is called for the anemia and beta thalassemia. So there is this specific kind of anemia, which is called sickle cell anemia.
Sickle cell anemia,
yes. So for what they do for the sickle cell anemia is that they as well, what they do, there is a gene that basically increases the level of the hemoglobin. It's called BCL11. Yeah, it's something irrelevant, but what it does increase the level of the hemoglobin. And then those patients so we can get from the normal people as well, we can get the hematopoietic stem cells, modify this gene that increases the level for the hemoglobin, and we can, by infusion, we can put to the patients that have this type of the anemia and beta thalassemia, and what it happens that then the levels of the hemoglobin goes up. And those, what they have seen, basically, I'm not sure if this is really like now approved, but is in the clinical trial. So these studies that I'm talking about are the clinical trial studies. They could see that the patients, after a couple of months, can get 90% higher hemoglobin than that's normal. And this is very, very important because there are therapies for those kind of patients are so limited that this basically can revolutionize this kind of, basically what we do, we disrupt this gene, and this gene goes where the hemoglobin goes up. So it's this gene therapy with CRISPR looks very, very promising nowadays.
Neat. Could you tell us, in a very easy way, I don't know, very summarized thing, which are the main limitations, the main difficulties that this type of approaches may have as
Yes. Yeah, of course, as every approach is in the clinics, and of course, what it has its own downsides, right? So what it happens is that CRISPR has something what is called the off-target effects, okay? So meaning that it's not always targeting where it's supposed to be, like if we want to target this specific gene, specific region, this specific mutation, but sometimes it goes into the genome and randomly we can get some deletions, insertions. So and sometimes they have also observed that we can have like large deletions that can be quite toxic. Okay, but what is now, there is a lot of studies which are now that we have Carme can tell you more about this, but there is these different types of the CRISPR of the Cas9s, okay, these different forms of the Cas9 that are now have been tested. Yeah. That can be like more, let's put it in this way, so they can be more better, they can have less off-target effects.
Exactly. Also, the point is that CRISPR is so effective that sometimes it cuts to a similar sequence, but it's not the intended sequence. So now the field is moving towards making CRISPR-Cas versions that don't really cut the DNA. Just they sit on the DNA and then modify the expression, enhance or repress the expression of the gene of interest, but they don't cut the DNA, so they are less toxic and they are having less of these off-targets. And also the delivery of the methods, they also now, the field is moving towards using complexes, which are just not DNA that can be around, the CRISPR tools around there for like many, many times. It's just that the protein and the complex is just for a few hours there, does the job and disappears. So trying to solve this limitation of the off-targets, so there's a lot of work
there is an off-target effect, but also there is something what is called as well immunogenicity, so not only that we are now working towards of the Cas9, that to make them like to have the off targets, because we know it's apparently in our body, we also have anti-Cas9s, no? So what we need to make is the Cas9 to be less immunogenic as well. So this is all this what Carme was explaining, other new variants that are now have been explored to be the orthologs of the Cas9 that can be used in the clinic. So this is also one of the things. And the other one is also what have been described is that if you, it's kind of a off-target effect, but yeah, because what it does as well is induces the when you have the CRISPR, it goes inside, so you have double-strand breaks. And double-strand breaks, what it does, activates the p53, one of the genes that we did as well in the practice. And then this leads to apoptosis, which is a cell death of the cells. So nowadays as well, it's like, are we going to use only CRISPR in a p53 deficient cell? So this also, for the cancer biology, this makes a lot of, there's a lot of also strategies how we can overcome these kind of limitations, but still there is a long path for that. And what else I can tell you? I can tell you that, yes, also the infection with a Cas9, because Cas9 is a huge molecule, right? So to get inside of the cells, it's very difficult, right? So the percentage of the infected cells, what we call that, the cells that will receive the Cas9 is very low. So the efficiency of the Cas9 is quite low, but yeah, so nowadays, again, so there are these different types of the Cas9 which are smaller, so we can put them better into the cells.
They call it the mini Cas.
Yeah. Yes, that mimic a Cas9, yeah. Mm-hmm. So yeah, so there is like a lot of research that we are, that are still to do and make it better, but still, yes, it has its own limitations, but the people are working on towards very rapidly.
Good. Great, amazing. Okay, so great power comes with great risk as well. So Alberto, I wanted to ask you, which ethical controversies you see in a tool like CRISPR, I mean, should we regulate? How much should we regulate? What do you think about this, Alberto?
Well, yeah, as I really like your quote from Spider-Man, with a great power comes a great
Maybe I know.
responsibility. Okay. And here there are mainly two topics. So we could talk about applying CRISPR into humans and also applying CRISPR into other beings and the consequences we could have for ecological aspects. But right now, I would like to focus on what happens when we are applying CRISPR into human. And for this, I'm bringing you two different stories. So the first one, it's more academic and, let's say, serious and sad. And the second one, I consider it's funny and scary at the same time. So let's just start with the most academic one. On 2018, there was this researcher named Janic that he performed CRISPR experiments into human embryos. This was done without the knowledge of the ethical committee of the organization he was working with. And what he did was to edit the embryos of two girls, and the resulting human beings were missing one protein, which is called CCR5, which is the receptor that the HIV virus uses to infect T lymphocytes. So in theory, the resulting girls, here you can see them, were immune to HIV. However, reality is not that happy, because for one of them, this change in their genome happened in heterozygosis, meaning that only one of the two copies of these genes was actually modified, and the other had this modification as a mosaic. So this meant that some of her cells didn't have this gene, but some others, they did have it. So at the end, these two little girls had been exposed to CRISPR and all the dangers that it involves, such as the off-targeting effect that Anna was saying before. And at the end, none of them got an effective protection from HIV. Since all this process was done without knowledge by the scientific community, the scientists who conducted these experiments now has been banned to perform research anymore. And actually, I think that he has been into prison for 3 years. So this is like the first story where we see what happens when the power of CRISPR falls into professional hands that use it a little bit irresponsibly or without regarding the ethical concerns of the whole scientific community. We have to understand that, science works as a community, and going on your own, it's not going to help you. But then we have...
Before you go to another story, what do we know about, because I remember the story on the news, but what do we know about these girls now? Do we know about possible problems or?
As far as I know, and here if Karma and Anna want to help me, like, they are super welcome. As far as I know, there is they have not developed any disease, so we are aware that the protection against HIV is not effective, but we are not seeing any disease that we could expect derived from an off-target effect from CRISPR.
So all the possible mutations that they probably got, they did not harm them so far.
We cannot see if the mutations are there or not. We are seeing that they are not appearing as in the form of a disease. Here, what I have in mind mainly, it's what happened with the gene therapy experiments that were conducted first on the 90s. And for some of them, there were many cases of leukemia associated to the treatment of several genetic conditions. And that stopped completely the development of treatments in gene therapy until very recently. So here, what I think it has to be highlighted is that CRISPR, as well as any therapeutic that comes with gene editing, it comes at a risk, and you're playing with the genome, and we don't fully understand what all the parts of the genome are doing. So if you edit the genome, you are taking the risk of maybe developing or increasing your likelihood to have cancer or neurodegenerative disease in the future. That is why if you are taking this treatment, it should be to treat some conditions that are having a strong impact in your life. So you're having a high risk treatment, but it also should be used for conditions that if they are solved, they are going to give you a high benefit. And therefore, here Anna was talking about cancer, was talking about sickle cell disease. But the problem is when the relevance of CRISPR is somehow banalized, and then you start having people that want to use CRISPR to have blonde kids or to get like super muscular, which is the case of the second story. I don't know if any one of you wants to highlight some to make some comment, but yeah, CRISPR brings you one step closer to this idea of designing your kids. But at the same time, I think that it's really dangerous, and I think it's extremely stupid to put your kids into the danger of having off-target effects just in exchange to, being blonde or having blue eyes.
At the moment, this is not possible. This should not be possible because it is forbidden, at least in many countries, to touch germlines or zygotes, or right.
That's right. It's completely forbidden. But
I don't know if it's in all countries. I suppose there should be some gaps.
Yeah, in general, this is forbidden in all countries, but some of them have more permissive guidelines. So as far as I know, the experiments that I was talking before, they were conducted in China, and China has a more lax policy in order to work with gene edition and so on.
You wanted to tell us another story.
Another story. That's a
fun story.
That's a funny one. So on 2017, a video appeared on the internet about one scientist, properly trained scientist, PhD on molecular biology by the university
from the NASA, I think,
and former worker of the NASA as well. And the video featured this guy injecting himself DNA encoding for all the CRISPR machinery needed to turn down one gene, the myostatin gene. So before going into the details of what gene was he trying to turn off, I would like to ask Carme and Anna if injecting DNA in your muscle cells, just as in a movie or in a video game, if that could have some sort of effect.
Well, this has more dangers than the benefit that you can have, because probably it would be destroyed, and it can even be immunogenic, as you were commenting before, or Anna, about these first genetic experiments with gene delivery. So I think it's pretty scary to do that.
It's not something that you should do at home.
Oh, definitely. But then, because this is something that we've been commenting before, in order to deliver the CRISPR machinery inside the cells, they are using either virus or particles, but never the DNA on its own. So this takes us to the second point. So just in the hypothetical case that this procedure was an effective form to deliver Cas9 and the guide RNA inside the cells, let's take a look to the gene that he was trying to edit. So he was trying to knock out a gene that is myostatin, that it's a gene that is inhibiting muscle production. Okay, so for those of you that are listening, I recommend you that go into Google and search myostatin deficiency cows. So this is a strand of cow that appeared just by several years ago, not by genome editing, but just because of farming. People were selecting cows that were able to produce more meat, and at the end they found out they came up with these cows that they are huge and they have huge muscles, and that's a lot of meat. So the guy was trying to do this with himself. And you can think, okay, I guess it's cool having huge muscles, but then you think about the power of this technology and also the off-target effects. So you are risking yourself to develop leukemia or other disease in exchange for having big muscles. So that's, for me, like it's funny at the same time that it's scary.
Yeah, actually, it's a bit the same thing than with the HIV. With these babies that were modified the CCR gene, because there were alternative treatments to prevent infection, right? So it's a bit overkill to use this when you have other possibilities. So,
and even that, this experiment of the CRISPR babies was not a successful one in terms of technical. So, like, the babies are mosaic. So it means that since the CRISPR tools that these Chinese scientists used were the first tools that are not refined enough, they are not developed well enough, they took time to start editing the cells. So the embryo had divided several divisions, and then some cells edited and some cells didn't. So in the end, the girls, some cells are edited and some are not. So not all the organism is protected. And moreover, so we have 2 copies of a gene, right? So this, one of the girls was heterozygote. So it was just 1 copy was edited, the other was not. So in the end, they didn't achieve those, which is not the way to. There's other ways to avoid HIV in the embryos, but by this technique, he didn't achieve that. So it was a hard job.
Good. So I think we commented when we introduced the technique. We thought that we discussed a bit on the directions, on the ethical aspects. So if anyone is listening to us, hopefully yes. We are open to receive questions. So please drop us a message, and we will read it and introduce to the discussion. So maybe while we wait to warm up a bit, you mentioned before these modifications of the Cas9 that are is ongoing work just to make them smaller or more tolerant.
Yeah, there are many versions of Cas proteins now. So some are already natural versions of Cas from other bacteria that they have other sequence limitations. So their PAM sequence is different. So you open up the possibilities of design of Cas. There's also this mini Cas, which is like 4 times smaller, so improves the delivery, which is, apart from the off-target, another limitation of the CRISPR is the delivery. So by the smaller the thing is, the easiest to deliver into the cell. And then other modifications are like engineering of the Cas9 protein to make it enhanced, like more specific, like have less of targets. So there's the hyper-accurate Cas9 or enhanced Cas9, and they're all going into this direction of having more specific Cas9 proteins. And also the mutant Cas9. So this is a Cas9 that binds to the DNA but cannot cut it. So then if you fuse this Cas9 to other proteins or domains which are DNA enhancers or repressors, you can enhance the expression of certain DNA regions or repress it. So all these modifications comes from like the original Cas protein, and this is like increasing and increasing. And another, I want to also point another Cas version, which are called Cas13 and Cas12, which instead of recognizing DNA, they recognize and cut RNA. So there's applications that have been developed to detect the presence of a certain RNA in a sample. So for instance, there are kits for the detection of COVID, of SARS-CoV-2 virus in a sample, which are CRISPR-based. And this is cheaper and more specific and faster than like the regular antigen tests that we know. So these are called Sherlock or detector, depending on the lab that has developed similar technologies using equivalent Cas12, Cas13. So they recognize RNA.
Sorry, Carme, these Cas12, Cas13, are these engineered proteins, or are these
No, they are natural proteins.
They are natural proteins. Are they homologs of the Cas9 or not necessary? Do you know?
Well, so the CRISPR system exists in bacteria, in 50% of bacteria, and archaea, so like very ancient species, and they have been existing here since millions of years. So there's like a broad range of different Cas proteins which are already there. So systems like this, in this war in between virus and bacteria, the bacteria has coming up with different solutions to.
So they originate from a common ancestor. All of them?
Sorry?
Do you think they originate from a common ancestor, all these Cas proteins?
Yeah, probably. Yeah.
Good. Okay. Also, it is nice to know that. This Cas became very famous, no, for the easiness of in use, but there were other proteins that had been used before with the same function, right? So the zinc fingers, the talents, right? These are also nucleases as well.
So they were the previous versions of genome editing, but they were like super cumbersome to use because in the CRISPR system, the nuclease, the protein is always the same, one Cas that you choose. To design the specificity, you have to just engineer an RNA fragment, which is very short, and this is easy to develop in the lab. But the zinc fingers were like proteins complex that you had to design specific protein for specific gene editing. So only biologists with expertise in protein design, they could do that. So they were fusing several zinc finger domains. Each zinc finger were recognizing 3 base pairs, so you end up with a protein that you had to engineer for a specific sequence. And this is very difficult. And talents is like 1 domain per 1 base pair, so there's 4 different talent domains that you combine. But in the end, the protein is huge, and it's also have limitations. So CRISPR bypasses all that.
Good, great. We have questions. So we have 2 questions. So let me read the first of them. First question from Adria. I have a question. Is it worth the hype about CRISPR? And if so, how do you think it will move forward? So Carme, Anna, you are
I can answer. So I think we kind of touched on this. I think first how it's going to move forward, we have to make it like more safer, of course, and we have to also learn how to deliver it much better, right? So this is very important. And what Carme was mentioning, that we need to like make now what we do is that we make these complexes between the naked RNA, what we call, then the pure protein, and then deliver inside of the cells, right? So what I think is nowadays is the most important how this will basically move forward. And of course, in the terms of in the lab, we in terms of the CRISPR screens and all the knockout and making like single mutation inside of the intron, exon, this is so fascinating. Then we can nowadays as you know, probably there is this kind of bioinformatics tools that we know that there is like millions of mutations, not only in cancer, but other diseases as well. But we don't know if these mutations are so important for the disease or not. But we can make this single point mutation and see if actually function is so important. So I think in terms of the biomedicine is also in the terms of research is very important, and it will move towards that. I don't know if I answered this question. Carme, please.
I fully agree. So if it's worth, it's definitely worth, because now many experiments or questions that could not be answered, now you can do it. So for instance, in our lab, we tend to do knock-ins. So we combine the CRISPR-Cas9 system with a repair template, in by which when you deliver these 2 things, you can introduce fragments of DNA in specific points of the genome that you want to study. For instance, fluorescent proteins that marks you a gene, but not only a gene. this can be used to mark populations, so relevant populations for cancer, which in a cancer there's heterogeneity, and there are cells which are like metastatic, others that maybe might be important for chemoresistance. And so what if you eliminate those cells? What happens with the tumors? So all these experiments, now we are able to tackle with the CRISPR technique that this was impossible. So this is, I'm telling just an application in the lab I work, but the limit of the CRISPR is scientist imagination, because every time papers like this is a boom of people thinking on new applications or ways to apply the CRISPR technique to solve questions and problems which are relevant.
Good. We have another question by Christian. Christian says CRISPR could be a good solution for the treatment of aggressive cancers. Yeah, is there any case of success in this direction?
So it's I think this what I have explained, how they basically in 2018 patent, well, actually FDA approved for the using CRISPR when they genetically modify the T cells, is actually used on the patients that have been highly refractory to resistant to the therapies. Okay, so I think this is like one of the most important experiments, well, the clinical trial that has been now performed, and this is for the cancer patients that are were resistant to the therapy, and this was used for the melanoma, multiple myeloma, and there is one type of the cancer more that I can't remember on top of my head, but that was used in this same clinical trial. It was successful.
Good. But CRISPR is patented. So what happened with this? Is it going to be a problem for future use?
Well, the patent thing has been a very polemic one, because like the Nobel Prize was given to the Untitled Chapter team because they were the first to publish a genome editing, CRISPR-based genome editing tool in a tube. So DNA mixed with these tools that they could cut the DNA. But then there was a parallel lab run by Ben Zhang and his mentor, George Church, and just months after, they published the first CRISPR-based edition in real cells, in vitro, but in human cells. So then they patented in the fast way, the second lab, and then the Untitled Chapter also, like, they were on litigation and trials to fight for this patent, and this has been taken like for 10 years. And it was resolved very recently, and they conceived the patent to the Zhang lab. But I think that even if it's patented, for research purposes, it's super open to everyone. So even the Zhang lab, they have websites, his website with servers to find guide RNAs towards any genome. You can also get their tools like plasmids and other tools. You can get them for free through platforms like AdGene, in which you can get regions from scientists. So this is the most used region in the AdGene platform is from Zhang. So like the research is open. So I think the patent applies more for like the commercial use of CRISPR as a therapy.
So do you see it as a problem in order to deliver treatments at a reasonable price, or?
Well, of course, I think you need to patent the advances to make it like to make a stimulus to the people to develop tools and make startups.
But what is your thought about the patents? I mean, patents like this. I mean, can we patent something that nature made before? I mean, we have not discovered anything new, right?
Well, this is like, okay, this is very technical, but the tool is already engineered because they call single guide RNA. It's what they are mixing. The tracer RNA, which is naturally separate from the guide RNA, so they make it fusion. So all these modifications maybe are not so obvious to make the genome editing tool. and then all the delivery systems, everything. I think you have to invest to develop all that. I don't know what you Alberto or Ana, you think.
I completely agree, and I really would like to stress out, not only in terms of like patent that is so important, is what we were talking, is that it's all kind of resources that are around and they are so affordable, right? So first, in the research base, of course, we're not talking about the clinics, because still we have to go through the clinical phase 1, 2, 3, right? But is this really made like our research not only in different diseases, and you could see, for example, it has already applications in COVID, and it's really amazing. And we are talking about like much cost-effective technique than it was before. In terms of the research, and probably in the future will come into the clinics. So in terms of patents, I think it's important, of course, we need to patent the technique, but will be very soon will be available to everyone. So, and also what she was saying, like Zanglab, that has all these resources for free online, and we can design our CRISPR guide with, there is so many online resources how you can do it yourself, which is for free. So you don't need to pay all these things. And this is what is beauty of the research, and how we can share our tools.
Yeah, I think if this particular lab wouldn't have shared so much, maybe the CRISPR would be not so broad in all the labs in the world. So they are also pioneering this.
So you're positive in this regard. You don't see.
Yes, open science research is so important, and the science is moving towards that.
Okay.
And then patents, they are not forever. So you can patent for some years, so you can recover back all the investment that you need to develop a product. Like you need clinical trials, and these are super expensive. So otherwise, new therapies wouldn't be developed, I think.
Yeah. Okay, another topic I would like to elaborate a bit more. I was also shocked by the video Alberto was mentioning of the guy injecting the CRISPR kit just to build his muscles. So the fact that the technique is affordable, I mean, the experiments are relatively easy, right? So anyone can buy these kits or do their own, right, Alberto?
Okay, so I have been doing my research, and I got into the website of this guy and of his company. It's called the Odin, by the way. And the kits that he is selling are not to edit your own genome. They are genes that come with population of bacteria, or in some cases, if you want to pay a lot of money, they come with animal or even human cells, but they are not intended to edit your own genome. They are intended to take some bacteria and do something similar to what you have done in the practical. So you take some bacteria, you transform them, you see how the CRISPR machinery modifies the genome of your bacteria, and at the end you see some colonies in order to check out that the gene editing has gone right. But in these kits, they are not selling anything that it's going to allow to edit your own genome. And even more, I really like the YouTube channel from which this Kutzegast, it's a great YouTube channel from which this image comes. On an interview they did to this guy, he strongly regretted about injecting himself DNA containing the CRISPR machinery, and because after him many people was doing the same thing, and he felt that that wasn't right, because although the DNA is not able to get inside your cells and promote the edition of your genome, as Carme was saying, it can be immunogenic. You're injecting yourself like foreign DNA into your genome, so into your organism. So that's never going to be good for anything. And he felt sorry because many people took his action as an example. and then he was regretting. So the thing is that all the things that are sold in his webpage are to do small experiments in your house, small experiments that indeed they are expensive. So it's way more expensive than making like a soda volcano or stuff like that. But at any point, he's selling stuff that it's going to enable people to modify their genome. And if he would, I guess that would be totally banned by the Food and Drug Administration at the US, at least.
Okay, so you think that we should not be scared so far about people playing, modifying its own genome? Good, but I mean, this is just speculation, huh? But I mean, if you are able to modify bacteria and these bacteria, just escape from your laboratory, they can go somewhere and replace the current population, right? So.
I think all these experiments to do by amateur people at home, I think you have to be very, very careful, because now you will say, okay, this DNA is not working in the delivery. Now it's not solved, so it's not working. But let's imagine that you have a kit that you sell to wherever for no purpose, people to genome editing their cells. I think this, of course, should be very tightly regulated and just for medical purpose, of course.
Yeah, and this is related to the question that I see here in the chat as well. So from Christian, so this is, I'm not sure, but this is forbidden, right? So those kind of people cannot do these kind of things, and ethical regulations are in place. So I would like to assure everyone who is listening to this that as a scientist, we have the ethical regulations, and very, very strict one. So for every procedure that has to be employed with a human cells, patient-derived tissue, with any model organism, any animal, it's strictly regulated and has to be strictly justified. So in Spain, and because Spain works as, according to the European Union rules, and there is a lot of administration and a lot of people involved in this ethics committee that has to be approved before any of the experiments that are touched by us. So these kind of things are actually, is what I see he's mentioning politicians like Trump. So this is all, I don't know what Trump can do at his home, but definitely not in the labs that are actually, official and governmental funded labs or industry as well.
Good. I heard a video with Vladimir Putin saying that he was scared about CRISPR. So I'm scared about this as well.
Maybe because he doesn't know much about CRISPR. That's why all these sessions are so important, that we can tell the people, that they shouldn't be scared about what we do in the lab and, the potential that it has, as long as it's controlled. And I can assure everyone that it's tightly controlled, at least in this country.
Okay. So I don't know, maybe you could say something on applications in agriculture, or for instance, in controlling, malaria. I think there is a nice example with CRISPR.
Yeah, so there's an application in which they plan to combine the CRISPR knockout technique, genome editing technique, with a gene drive technique, in which you can, with the idea of manipulating the inheritance, so the reproduction of vectors for malaria or Zika disease, of mosquitoes that can carry diseases. So try to impair their reproduction by this system. So this is also risky, because you start moving to the ecosystems field. So something that might be nice in the paper can get out of control. So this is not my expertise, so I don't know the cost benefit of this, but it has to be also very tightly studied before.
Yeah, but this is the direction that I wanted to open. I mean, these things may have consequences, And so, do we know enough? I mean, in biology, in ecology in particular, to be able to go ahead with these things? Because I mean, if we remove the mosquito from an ecosystem, I mean, do we know what's going to happen, or can we predict?
Yeah, well, previous experiments with trying to play with the ecology end up bad. That sometimes there's like something that escapes, that acquire resistances, or like, to me, it's scary, to be honest. But it's also like, it's true that malaria is a real problem, so it's difficult to deal with it.
Yeah, I mean, if you are in a country where you have, the probability of getting malaria once or twice a year on average, I mean, maybe you want to have this risk, right?
Yeah.
Also, in this regard, regarding the case of Crispr and malaria, what I have heard is that they are not planning to wipe out mosquito populations, but instead just having mosquitoes that have like the smallest gene modifications, so that they are not able to transmit Plasmodium. But that's going to be, in theory, the only difference. Here, what I find that it could be scary, it's maybe that's what it's written on paper, but maybe those mosquitoes have some increased fitness or decreased fitness in comparison with other mosquitoes or other organisms in the ecosystem, and maybe they can spread and come up with an overpopulation of mosquitoes, or the opposite, having a decrease the population of mosquitoes in the place. And of course, that plays a huge role in the maintenance of the ecosystem. So it's complicated.
Yeah, so this is something, when it's out of the lab, it's out of your control. So you don't know what can happen in nature.
It comes to my mind now. I think it happened with some algae that were tagged with a green fluorescent protein in some aquarium, and they broke out, and now you can find them everywhere. I don't know if you're familiar with this story. I was told this when I was in high school. I don't know. Okay,
No,
maybe my sources are not. It's just in case that you remember. Right now, I don't remember my sources, but somebody explained me that situations like this have happened before.
But I think you mentioned, Carme, before while we were preparing this conversation, that there are CRISPR modified goldfish.
Yeah, so this has been approved. Yeah, but I don't think they are released in nature. Maybe they are in. So in Japan, they have approved a CRISPR fish, a goldfish with Harrison mutation that makes this fish more hungry. So they eat more and they get bigger. So they are bigger fish. So maybe this is for production of fish for human consume. Yeah.
Good. Okay, so I think it has been a very nice conversation. We have been more than an hour online, so maybe it's time to stop. To start stopping. So I don't know. Do you think? Would you like to touch any topic that we haven't covered before? Okay, so if not, maybe I will just ask you a final question. I mean, are you excited about CRISPR? Are you scary?
Absolutely excited. Thank you for discovering CRISPR, what I can say.
No. I think we made our point clear since all the session that despite the limitations or dangers, the world is moving towards this. We have to accept it, and I think that there's more of a benefit and advantages than other things.
Great, Alberto.
Completely agree. So
great. Great. I mean, I don't know if I did right or not, but I watched this one of these Netflix documentaries, and I was very scared, actually, with biohackers everywhere. But after talking to the experts, to you, and now I'm far more optimistic. Okay, so thank you very much, Carme. Thank you, Anna. Thank you, Alberto, and thank you
Thanks for the invitation.
to everyone that joined. Hope you had a good time, and hopefully we can repeat this in another topic or again and talking where we are in CRISPR in a while. Okay.
Okay.
Thank you very much.
Thank you.
Bye-bye.
Thank you.
Bye.
Bye, everyone.
Bye.